Hello students, I am Dr. Ashwath. In this session, we're going to discuss about the transgenic approaches in plant resistant to bacterial infections. Generally, plants affected with some bacteria as a pathogen and causes many diseases here. Bacterial blight disease and root rot disease, some other diseases will be there. So bacteria successfully infects the plants, but plants also produces defense mechanism against that. But some bacteria will also go against and then escape from the defense mechanism and cause infections here. So here the transgenic approaches will give support for the bacteria. Definitely will go, scientists will go with the transgenic approaches so that they will effectively produce a elite plants which is resistant to multiple bacterial resistance. So these are the parameters and these are the strategies we can use in resisting the bacterial infections here. So these are the parameters here. So we can go anything here in transgenic plants so that we can easily combat the bacterial infections here. We will go one by one first. First one is called as cross protection mediated. As in previous slides, previous videos, we discussed that as compared to the as same as the virus infection, whenever the pathogen virus infects, that causes pathogenicity, right? Same way, we'll modify a bacteria in such a way that they produces milder infection, but it retains the antigenicity of the pathogenic bacteria. A milder bacteria, attenuated bacteria, will be infected with the plant so that plant successfully defend that pathogen and then memorize the antigenicity of that bacteria. And whenever the actual bacteria infects, definitely plant has this defense mechanism and then easily combat uh, the bacteria pathogenicity of the bacterium infection. This is called as cross protection mediated resistant approaches here. Next one is called as expression of antimicrobial peptides here. So these antimicrobial peptides, generally called as PAMS or AMPS or PAMPS or AMPS. So PAMPS means plant antimicrobial peptides. AMPS means antimicrobial peptides. Both are same. In examination, to confuse you, they will ask what is PAMPS. Same, plant antimicrobial peptides. These peptides derived from the plants that will secrete whenever pathogen enters into the plant defense plant territory. So if you observe that some proteins are there, these proteins are peptides which is released from the plants against multiple bacterial, fungal, as well as other type of viruses too. Here these are the, see here, this is one of the peptide, antimicrobial peptide. This is called defensin or defensin. Defensin is a small thionin or cytosine rich peptide that is released from the plants with respect to bacterial, viral and fungal infections. And if you observe that these are the defensin proteins that is produced from the varieties of the plants which is produced in Leguminosae family, that produce in the cabbage family, that produce in Solanaceae family, like the different family, even grass family, Poaceae also produces these defensin chemical against different types of pathogenic infection. You can see these are the defensins that has different activity. You can see these are the defensins which has antifungal activity that produce in different plant resources. And how this works, defensin will mix with the different types of chemicals inside the cytoplasm, and what happens whenever the pathogen enters, those peptides will be targeted with these antimicrobial peptides. So whatever happens, the bacteria contains genetic material as well as the cell membrane that will be disrupted because of the release of these antimicrobial peptides outside the damaged cells here. That is the defense in here. One more chemical you can see here. These are the list of genetically modified defensins that is produced. That means, these are the source plants that is produced, and then we introduce same genes, defensive gene, into the host plant, recipient plant, and they effectively combat the infection of the bacterial pathogen. Next chemical, antimicrobial peptide called as theanine. Theanine is also a small peptide rich with cysteine. You can observe that is also has a defense mechanism. Whenever a pathogen like fungal, bacteria, whatever enters to cytoplasm of the plant cell, what happens? So these will trigger out a reactive oxygen species. Whenever reactive oxygen species like hydrogen peroxide, H2O2, or nitrous oxide will be produced, that has the cell, a cell defense mechanism that will degrade the cell, pathogenic cell here, especially bacteria cell. So like that, this theanine will trigger out the reactive oxygen species and then helps in program cell death. Whenever bacteria enters, it will activate the reactive oxygen species, and then it will reach up the bacteria and then create a oxidative burst. Whenever oxidative burst is happen, that will trigger out the program cell death of that infected plant cell. One more, antimicrobial peptides, that is called cyclopeptides, that mainly secreted from Rubiaceae and Fabaceae family. So these chemicals mainly specific to some families of the plant, and even some plant defensin like proteins also, like that mimic like a defensin, but the composition is different, that will secrete from different plants, that is also has antibacterial activity. And thaumatin like proteins, these chemicals mainly larger than the defensin that produce from different types of orchid plants. And snakine peptides also there. They are also antimicrobial peptides, that is cytidine rich antimicrobial peptides are there, that mainly produce from the plants, that goes against with the fungal bacterial infections here. Next approach of bacterial resistant in plants is production of antibacterial proteins of non-plant origin. Take example, some chemicals will be produced in different organisms. Like you can see here, when a virus is there, that will produce a chemical. That chemical will defend the pathogency of the bacteria. Same with some virion. Same with some fungus. The fungus which goes in culture medium, which secretes chemicals that will defend the bacterial infection, as well as it can have a zone of inhibition between fungal colony and bacterial colony in culture medium. So if you take some proteins that secretes from a different organism like a bacteriophage releases some chemicals that can easily degrade the bacterial cell. So that's the way we can find a protein or a chemical that is produced from the bacteriophage that helps in degradation of the bacterial infection. Next one is antibacterial peptides, that is for either from the animal, fungal, insect, or bacterial origin, that can be extracted and then will be inserted through by transgenic approach into the host plant, and definitely the plant will produce that protein, and meanwhile that protein will degrade the bacterial infection, bacterial cell wall. And then even some chemicals are produced from the insects that has bactericidal activity, bacteriostatic activity. Bactericidal means that will kill the bacteria, and bacteriostatic means that will stop the propagation of the bacteria. And one more chemical, that is called chitosan, that is a biopolymer that is produced from the shrimps and crabs. These crabs and shrimps, whenever bacteria infection happens, it produces a chemical called as chitosan. This chitosan has antibacterial activity, that can be, this chitosan gene is inserted in plants, that plant will produces those chitosan that will defend the bacterial infection in plants. Even some silver nanoparticles, higher concentration of silver nanoparticles will also has the antibacterial activity. We can insert that in the plant, and definitely that will plant will defend the pathogenicity of the bacteria. Next, one more approach is inhibition of the bacterial pathogenicity or a virulence factor. Here we can trigger out the plant immune response in production of some secondary metabolites or polyphenols will be we will enhance in that production of the polyphenols or secondary metabolites in multiple folds, so that it can easily defend the bacterial pathogenicity. Even we can whenever plant is there, we'll mix in soil, we'll mix with natural microbiota or other type of biocontrol agents like pseudomonas and other type of bacteria will defend the bacterial infection further. So these bacteria will useful which cause useful effect for the plants, but also prevent the infection of the pathogenic bacteria. Like that, we'll also use a biocontrol agent like a bacteria, fungi, virus, which will defend the pathogenic bacteria that cause infection in the plant cell. Like, and same antimicrobial compounds will be produced, and other type of plant-derived vaccines are produced. Likewise, we can easily inhibit the bacterial pathogenicity. Next, one more approach is called as enhancement of natural defense mechanism in plants. So we'll modify a genetic some genetic modification should be there by transgenic approach. We'll insert more number of these pathogen derived genes, so that that can easily defend and express more these peptides. Expression will be more in multiple folds, and because of the multiple fold expression, that will prevent the bacterial infection. Some chemicals are there. Take example like in neem plant. So that produces azadirachtin. This azadirachtin will have these antimicrobial, antibacterial property. When you increase these multiple folds in other plants other than the neem plant, definitely, so these plants also effectively defend these bacterial infections. Even will also enhances the systemic resistance in plants, so that it can easily defend the pathogens. And next method, like priming. Priming means treating plants with certain compounds or like hormones or elicitors that will enhances the pathogenicity, sorry, the defense mechanism in plant cell. So a response will be faster with the plants, and then plant effectively mitigate the bacterial infections here. And even will increase the physical barriers, like cell wall lignin components will be secretion will be more. Whenever lignin secretion is more, definitely that will easily stop the penetration of the bacteria inside the cell wall. And even we'll also, one more approach is called as activation of defense pathways. Like we'll have a jasmonic acid pathway, other type of pathways are there. Will enhance the reactive oxygen species in plant cell, or activation of some protein kinases that will target bacterial cell. Like that, we can easily target the bacterial cell by changing the immunity and other type of resistance in the plants here. Like jasmonic acid pathway, ethylene pathway will be there. This jasmonic acid in plants will trigger the defense mechanism, and then whenever jasmonic acid, ethylene is produced, definitely whenever infected cells are there, they will be degraded, and then the transmission of the disease will not happen to healthy cells for. And even will target a callose. This callose is a enzyme that will degrade the beta 1,3 glucone linkages, and then we can enhance the production of this callose multiple folds, and then we can reduce the pathogenicity of the bacteria. And even will enhance the production of these chemicals called as phytoalexins, flavonoids, and glucose in nolates. These chemicals will definitely prevent the infection of the bacteria. Next approach means induction of programmed cell death in the site of infection. Whenever we change the genetic parameter of the host plant in such a way that whenever bacterial infection happens, so it will trigger out automatic program cell death. Whenever program signaling cascade is triggered out, definitely those cells will die instantly, so that the transmission of the pathogen to the healthy cell through plasmodesmata conduction will be prevented here. Even will enhance the hypersensitive reactions, even will enhance the antimicrobial compounds, and even will change the composition of cell wall through which bacteria enters into cell. So like that we can prevent the infection of bacteria into the things here. And one more approach is called as expression of bacterial toxin genes. These genes either originate from the bacteria or from the other closely relative bacterial strains. You already know that two different bacteria will not grow in a master culture plate. They have zone of inhibition because they will produce some chemicals that will defend the opposite bacteria. Like that, one of the chemical is bacteriocin. Bacteriocin is a chemical that mainly produced from some strains of bacteria, like E. coli and other will produce this, and even the lactococcus will produce as nisin. Nisin is a bacteriocin type of chemical which prevent the growth of other bacteria. So that same cross production mediated resistant or opposite bacteria which produces a chemical to defend the opposite bacteria that will be targeted in plants. Plant will be engineered in such a way that it produces these types of bacteriocin, definitely that will prevent the infection here. And T6SS, T3SS type of the chemicals also target the other type of bacteria. In Pseudomonas, you can observe that these proteins, which mainly inhibit the growth of the other competing bacteria that grows in culture medium. And even one more chemical called antitoxins that produce from the bacteria, especially some E. coli that produces these type of chemicals that prevent the propagation of Mycobacterium tuberculosis. Hemolysin and sensing controlled toxins are there. They are the chemicals that produce from the opposite bacteria that prevent the growth of opposite bacteria that grows in culture medium here. And one more type of approach is called as introduction of pathogen derived resistant genes. You already know that in some agriculture crops, so these resistant genes are formed because of the severe infections. Like you can observe that XA genes. This XA genes, these genes will which is resistant to Xanthomonas oryzae that cause bacterial blood disease. These genes are introduced in different plants which have similar infection of Xanthomonas, so that we can go for transgenic approach in different plants that will prevent the infection of this Xanthomonas pathogen in different agriculture crops. Same with tomato, same with pepper, some other type of pathogen derived resistant genes are introduced so that we can easily prevent the infection of the bacteria. And next approach is called as RNA interference mechanism. You already know that whenever the bacteria circular DNA is enters, what happens, some part of the genes will be targeted, and then these genes will be introduced as an antisense RNA. So this antisense RNA of the entire strand will be introduced into the plants whenever the sense strand of this pathogen will integrate into plant genome that will express and produces the mRNA. Same way in plant also, because of introduction of this antisense RNA, there is also produces antisense mRNA. This antisense mRNA and mRNA will, because of the complementation, they will form a double-stranded mRNA. This double-stranded mRNA in plant cell is not natural. So that will be recognized by the plant defense mechanism called as dicers. Whenever dicers will recognize that, that will trigger out a risk complex, risk complex, RSC complex will be generated. This risk complex will recognize that and then degrade the double-stranded mRNA that produces. Otherwise, one more mechanism is that it will be recognized by nucleases. These nucleases will degrade that completely. This is the way we can target the bacterial genome that will not express because of the formation of risk complex and degradation of the target mRNA of the bacteria. Next, the well-known mechanism, you already know that in the CRISPR-Cas9 type of genome editing technology, we will target the bacterial genome. Some protein will be targeted that is essential for the expression and propagation of the bacterial cell inside the host organism. Will target that protein, and then because of the production of that protein, so bacteria cannot propagate, and then that become uh, toxic, and then bacteria instantly die in the host plant because of precise genome editing technology like CRISPR-Cas9 or talent technology or other technology precisely edit the genes and then prevent the bacterial infections. And one more method is called as lysozyme mediated bacterial resistant. So lysozyme is a enzyme mainly present as a defense mechanism in phagocytosis process. What happens? Lysozyme targets only bacterial cell that mainly breaks out the beta 1,4 glycosidic linkage between N-acetyl muramic acid and then N-acetyl glucosamine that is mainly present in the mucopeptides of the cell structure of bacteria. That will only target that. These lysozymes that mainly originate from different type of bacteria will be integrated into plant genome with the a strong promoter. That means cauliflower mosaic 35S promoter will be introduced. Whenever that will happens, definitely expression will be more and produces these lysozymes. These enzymes, whenever bacteria infection happens, will target the bacterial cell and degrade the cell wall, cell membrane, and then bacteria because of the cell lysis will be died in the genetically modified transgenic plants here. This way, we can easily neutralize the bacteria. One more approach is lytic peptide. In examination, this will be asked. This lytic peptide generally a amphipathic. Amphipathic means they will act like a positive and negatively charged uh, chemicals. Whenever whenever acid is comes, it will become new basic. Whenever bases will come, some it will become acidic. Like that, it is called amphipathic. Alpha helical peptides are there. These peptides mainly targets the gram positive and gram negative bacteria. Here, some of the peptide is very important for you that will mainly produce from the different insects that resist the bacteria. Take example here, sarcopine mainly secreted from the silkworm. It contains 26 to 25 28 amino acid that mainly resist the bacterial infection. Remember, so when you introduce these sarcopine genes into the plant cell, definitely plants also produce this sarcopine chemical and then resist the bacterial infection. One more chemical is called melittin. Melittin also mainly extracted from honeybee. This honeybee will secrete that against the bacteria, and when you introduce same genes, definitely plants also produce this. And then CAMA fused proteins. This CAMA fused proteins, it is sarcopine and melittin fused proteins are there. In combined resistance, we can target that when we introduce sarcopine and melittin together. So effectively, we can defend many bacterial cells here. And Tassin mainly extracted from some moth pupa. We can observe that can has bactericidal activity that prevents the infection of the Erwinia kertovera. Thionine is a chemical that mainly produced from the barley and wheat plants, and we will introduce that to many other plants that can defend the infection of pseudomonas type species. And bacteriopsin, these also chemicals that mainly has defend against same pseudomonas syringae bacteria. And we will also enhance the reactive oxygen species in plants by introducing these genes in different places and easily neutralize the bacterial infection. So next mechanism is called as detoxification of pathotoxins and alternation of target enzymes. So in that one of the this mechanism means these toxins, whatever the toxin that is produced from the pathogen will be targeted. So same toxin that is produced from the bacteria in their body, in their cell, they have the neutralizing mechanism, remember. So the toxin will be neutralized, right? You see, whenever, take example, tab toxin is mainly released from a pathogen. This pathogen called as pseudomonas syringae. Whenever this tabatoxin is produced, this toxin will be dangerous for the plant, but it is not dangerous for the self bacteria. Why? Because they, in bacteria, there is a neutralizing mechanism is there that will neutralize the toxicity of tabatoxin. So whenever the some compounds of these tabatoxin will be introduced in the plant cell, what happens, especially in tobacco? So these transgenic tobacco will have resistant with respect to the same pathogen. By whenever we introduce a gene with a strong promoter, cauliflower mosaic virus 35S promoter will be introduced along with the tabataxin expression gene into the host plant, definitely that show resistant to some wildfire diseases that happens because of the pseudomonas syringae infection. So these are the mechanisms, different way we can modify a plant into transgenic plant and effectively resist the bacterial infection.